Hi, in this video we are going to look at the process of protein synthesis. Okay, so we will just look at your video and discuss some of the nuances that are not clarified in the video. So this is a cell. So if you know the cell, although the cells are different shape, if you notice the neuron has a different shape, but the cells have different parts, different organelles. And if you look at the central part, which is the nucleus, you have there inside the chromosome, and a chromosome is just again a package of histones and DNA. So it's coils upon coils upon coils of DNA. So the DNA is coiled around histones. The histones are coiled into uh, bigger and bigger structure until they become the chromosome. So this is the DNA, and then you have a segment of the DNA is a gene, right? So again, we have to go back to our analogy here. The whole DNA, the complete set, is like a set of encyclopedia for all the instructions of all protein, of all the processes in the cells and protein production in the body. And if you go to a particular chromosome, that's like going to a particular volume or a particular book in the encyclopedia. And if you look at a particular page in the book, like page 20 to page 40, you're looking at a particular segment in the DNA, and that segment is gene. So, say for example, if it's an encyclopedia of recipes, and then the recipe that you want to make, in this case, the protein you want to make, in the analogy, it's the recipe you want to make is making a sauce, a marinara sauce, for example. And let's say that recipe is in volume number 5, page 20 to page 40. That means in the DNA, if it's this certain protein, protein X, for example, you want to create that protein, and that protein instruction is in chromosome number 5 in this segment, that is called a gene. That set of instruction, page 20 to page 40, that certain string of instructions, that is a particular page in the book, in the particular in the encyclopedia, that is what we call a gene. Okay, and a gene is again a set of instruction for a particular protein, or it could be a particular segment of a protein, because later on in the fourth quarter you will know that the protein could have quaternary structure. So meaning a protein could have segment 1, segment 2, segment 3, segment 4. In fact, hemoglobin is like that. And those 4 segments are, when they connect together, they form hemoglobin. So something like that. And it could be that segment 1 is created from some other chromosome, segment 2 is created from some other chromosome. But again, in terms of analogy, it's like that. It's a particular instruction for something, or for a particular recipe. So when your cell wants to, for example, let's just say insulin. Insulin is a protein, and the body wants to create insulin. So therefore, that segment, that gene segment of the DNA that tells you the instruction for creating insulin is being read. It will be read by the RNA polymerase. Okay, and what the RNA polymerase does is the same as like the DNA replication, which is in the DNA polymerase itself. In the DNA polymerase already, um, it's already included there that there is some mechanism to split it up, just like the helicase in the RNA polymerase in the DNA replication. But it splits it up the same, right? And then what it does is it sucks in RNA bases and then base pairs with it. And important, when this RNA strand is being built, it's just like the DNA polymerase. It can only be built from the 5' to 3' direction. It can never be built from 3' to 5' direction. Okay? Which means that if this is the DNA, when you split open the DNA, remember, one end is 5', the other end is 3'. So if that is 5' to 3', so this must be 3' to 5'. Meaning, if you base pair it with RNA, if you, just like in the video, if you go from left to right, if you start here, then you have to build that strand, and since this is the 5', this is 3', so you have to go 3' to 5'. But again, that's not available. The RNA polymerase, just like the DNA polymerase, it cannot do 3' to 5'. So meaning, instead of building it here, it doesn't build it here because it cannot do this. It cannot do 3 prime to 5 prime. So what it does is it instead builds on the other strand here. Because if it attaches here, guess what? This is the 3 prime end, so this must be 5 prime end, 3 prime end. So therefore, if it builds this, it goes from 5 prime to 3 prime, which is the correct direction. So I guess what I'm trying to say is, sir, what determines which strand is red? Is it this strand, the upper strand, or the lower strand? How would the RNA polymerase know that it should be reading the lower strand and not the upper strand? The answer is this limitation. If it reads the upper strand, it has to go 3 prime to 5 prime, which it cannot do, so it has to read the lower strand. Obviously, lower and upper doesn't mean anything because the DNA could be floating in different directions. So what I mean by that is in this drawing, the upper strand happens to be 5 prime to 3 prime. So when you build this strand here, it must be 3 prime to 5 prime. It cannot do that. So in this configuration, it must read this strand, right? That's the idea. So subtle difference here between RNA and DNA replication, RNA, and protein synthesis. Number one, in DNA replication, both strands are being paired, right? But in protein synthesis, only one strand is being paired. And second of all, in the DNA replication, what is being base paired are also DNA nucleotides. But here, if you notice, the color of the strand, look at this color is darker compared to this one. That signifies that this is a deoxyribose phosphate backbone. But this one is ribose phosphate backbone. So meaning these nucleotides here are RNA nucleotides, not DNA nucleotides. It's a nucleotide made up of nitrogenous bases, A, instead of thymine, uracil, AUCG. And then the backbone is instead of deoxyribose and phosphate, it's ribose and phosphate. Right? So that's the difference. Okay? Then after that, sorry, I have to remove this and then play. After that, once that whole segment, that whole gene is being read, notice that the strand is not sealed. Unlike in DNA polymerase where after you base pair, this is sealed by the ligase to make it completely bonded with the other strand. This strand will simply just float away. Right? Will just float away. And you now create what we call a messenger RNA. Okay? And another difference, which is not shown in the video, is that the DNA is being split, right, into two. And then one of it is being read, one strand is being read, and then this one floats away. And then after that, the RNA polymerase will seal this back. It will seal the DNA back such that it will just become the old DNA that it is. It doesn't really undergo any change. So what I mean by that is it just temporarily opens this in the replication fork or an equivalent in the protein synthesis process. And then after you have read the DNA sequence, it will also seal this back in so that it will just become the DNA polymerase. And again, that makes sense. It's just like reading a book, right? You go to an encyclopedia, you simply open the book, you get the information you want, and then you close it right away. Right? It's not like in the DNA where we really split open the book and then build the other half and build the other half. I mean in the DNA replication. Okay, so now what happens now to that? You create an mRNA messenger. An mRNA molecule. This is what you have created from that. Right? And then now, I guess it's the time to to go further and discuss that in DNA in protein synthesis, which is what we are discussing now, there are two sub processes. The first sub process is called transcription. So this transcription happens from you read the DNA so that you can create an mRNA. So that process is transcription. So what we have just discussed, which is from the start of this, where it splits and then it base pairs it with RNA nucleotides, not DNA, but RNA nucleotides, and then again it's not being sealed; it just floats away. It just floats away from the DNA, and again the DNA is sealed back in. You now create an mRNA. So obviously it's an RNA because you are using RNA nucleotides, and it's called messenger RNA, so mRNA. That whole thing is called transcription. Okay. Now, why is it called transcription? So when you say transcription, transcribe, you're copying something, right? So think of it as, again, in our analogy, the DNA is like the encyclopedia of instructions, recipes of all the food items that you are ever to create. The problem is that encyclopedia cannot be taken out from the library. Think about it as it's really in the nucleus, and you cannot take out the book. You cannot say, okay, I will go to the kitchen, I will make a recipe, so I will bring this book with me to the kitchen so that I can glance over it and follow its instruction. Nope, the DNA process or the protein synthesis process doesn't happen like that. There are strict rules. One of the strict rules is that the DNA must be kept in the library at all times. You cannot take it out. So, what do? If you're a chef and I can't remember the steps because I don't have photographic memory, I have to copy the instructions into another piece of paper, right? So I read the instructions and then I copy it on a piece of paper, what the instructions are. Then once I have the piece of copy, I go now to the kitchen. In that sense, I have the instructions with me, the copy of the instructions, and I can go to the kitchen, make the food item, but I did not have to bring the book with me. The same thing. So when you say transcription, you really literally you're saying you're copying the recipe into another piece of paper so that you don't have to bring that book to the kitchen. You just have to bring that piece of paper. So the same thing here. Instead of bringing the whole DNA to where you are making the protein, you're just making a copy of the relevant instruction. Because if you think about it, you probably only need a few pages of that book. You don't need the whole book. So it would be more efficient to just copy it on a piece of paper. And in that case, that's the mRNA. Okay? So once you've copied that, there's a weird thing that happens, and this is what we call splicing. So what happens is once you have copied that piece of paper, some parts of it are cut out, and then some parts of it are spliced. Okay? What do you mean by that, sir? Meaning once you have copied the whole information, it turns out that some of it you actually don't use. So some of it you cut out and then you splice. What do you mean by that, sir? I don't really understand. This is now where I will have to remind you of exons and introns. These are segments in the gene. Exons are the quote-unquote useless part of the information. This is the gibberish part, the part that doesn't make sense, while the introns are the useful part. Okay? So, let's revamp the narrative. Think of it as the DNA is an encyclopedia of recipes, but those recipes are kept secret. So these are secret recipes of a restaurant, and you don't want the competition to copy your recipes, right? Again, this is just a narrative that we can think of. So if you think about it, if it's a copy of secret recipes, you have to safeguard it. So you have to safeguard it. You have to put it in a vault. In the previous discussion, I told you that the book should be kept in the library. In this case, maybe not even in the library, but in a vault, a secure vault. So therefore, you put you put the books in the secure vault, the instructions for all the recipes, and then when you put up a food item and you want to know the recipe, you go into the vault, you open the vault, you read the instructions of the recipe, and then you put back the book again in the vault, seal it up, and then only copy and only have the copy out, right? And that is actually what's happening in the cell. Why? Because think about it. The DNA is inside the nucleus, right? So what you do is you copy that DNA. Then after you have copied the DNA into an RNA, the mRNA comes out of the nucleus. You see, that's the nucleus. So the books, the DNA, are only stored in the nucleus. So think of the nucleus as the vault, the safety vault, or the safety. It's the vault where you keep your secrets, right? And you only copy a piece of it, and then after you have copied, you leave the vault to read the instructions. Okay, that's the idea. But again, I want to make this kind of like narrative in my head. Think of it as, again, the recipe are kept secret. So therefore, even if you copy the instructions, think of it as there is still a certain code. Think of it as like a coded message. So when you read something, for example, when you read something, if you have the message already, it's not the complete message. There are still hidden messages in it. So what I think is happening, or at least in my narrative, is like when you copy the mRNA, not everything in the mRNA is useful. Some of it are useless, called the exons. Some of it are useful, the introns. And it's only really the introns that matter. The exons doesn't matter. The exons do not matter. So you cut out the exons and you only keep the introns, because the introns has the instructions, the useful instructions. Okay? And again, why is it like that? Nga nung na may recipes or and then there are some things that are useless, some things that are useful. Again, think of it as maybe a way to protect against enemies. Right? So think about, I mean, again, indulge me in my story for a second. So for example, you're making a leche plan, right? And then the leche plan, there's instruction there. Number 1, get eggs. Number 2, mix the eggs with sugar. Number 3, add soy sauce to it. Number 4, add salt to it. Number 5, bake it inside the oven, and so on and so forth. So that's the whole set of instructions, right? But then, not knowing that instruction number 3 and 4 are actually not followed. So the instruction 3, which is to add soy sauce, and then number 4 to add salt, those are, think of it as decoys. Those are just decoys. Those are just instructions that are put in there to confuse the enemy. Because if the message is ever intercepted by the enemy, right? If the message is ever intercepted by the enemy, it might be that if they follow the instruction, they will still get a crappy tasting leche plan. Because again, the recipes are kept secret. So think of it as the DNA makes a copy of the recipes, but the copies of the recipes, there are actually parts there that are not followed, and there are parts there that are followed. And those parts that are not followed are called exons, and they have to be cut out from the mRNA, and the introns are kept inside. So exons outside. I'm sorry, I totally got this wrong. It's the opposite. Exons are the useful ones. Sorry. Exons are the useful ones, and the introns are the useless ones. And the way I knew that I was wrong was because of the word X. X means expressed, meaning these are the parts of the segments that are read, that are really followed, that are expressed into proteins. These introns are the useless bits. Again, and one way to, if you again think about virology, one way to think about exons and introns are Introns are decoys. Those are segments of the genes that are not actually used, but we still copy it just to confuse the enemy. And the enemy in this case are the viruses, because the viruses hijack our DNA and our protein synthesis machines. So that's one way to think about it. Another way to think about it is, these are buffer areas. Buffer areas, meaning what they mean by buffer areas, because it could be that um, mRNA number one is read from chromosome number two. Then you have mRNA number two. mRNA number one is read from chromosome number two, and then you have mRNA number two read from chromosome number four, and then you have mRNA segment number three read from chromosome four. And then, what if this is just part one of the instruction, part two of the instruction, part three of the instruction, and then you need all of those three to merge together. So you need to follow instruction number one, then number two, then number three to make a complete recipe. And what happens is this one and two must be merged. But again, you have a buffer zone here, such that when you merge these two, you have to cut this out first, cut that out, and then before you merge it, and then cut this out before you merge it here. So the machinery gives you some buffer zones that you can cut out safely before you um before you combine the three instructions. Because again, this mRNA instruction, they could also be from different chromosomes, okay, but that's the idea. So again, think about that narrative. The narrative is that the cell is secretive. So secret, it's very it has a lot of secrets and secretive, and it keeps its all the secrets in the DNA. And so when you copy, even when you copy, you have decoys and you have buffer zones, so that when you combine them, you can afford to throw some parts out. And those parts that are thrown out are called the exons. Again, so let me repeat here. You have the whole DNA copied, the mRNA copied, but some parts are kept out, and then some parts are joined together. So we call this splicing, this splicing part. And then now this now will come out, will move out of the DNA nucleus, and then we'll go to a specific site in the cell, in the cytoplasm. We're not talking about the cytoplasm here because it's now outside of the nucleus. Remember, the cell has a central structure called the nucleus, but outside of it is the cytoplasm, and the cytoplasm floats all the other organelles, including this next organelle, which is the ribosome. Um, so it floats outside, and then that mRNA is met by the ribosome, the large ribosomal unit and the small ribosomal unit. And then now the ribosome reads the instructions. Okay, it reads the instructions. And how does it read the instruction? Well, as it turns out, the DNA bases are organized in what we call codons, right? A set of three base letters. And the codons can pair with these three, which are called anticodons. Okay? So this is now the codon. So your mRNA has letters A, C, T, G, blah, 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 in a certain pattern. And we group that into three groups. The mRNA has what we call codons. Okay? A codon is three, like TAG, that's one codon. Right? It has a codon. And then what happens is the ribosome pairs it with, the ribosome pairs it with a tRNA. And that tRNA has an amino acid, and it has base pairs also at the bottom. And these here are called anticodons. Okay? So the mRNA has codons, the tRNA has anticodons or anticodons. Okay? And how does that work? Well, when we play it here, they base pair. So you see, C and G gets paired with A, C pairs with G, A pairs with U, G pairs with C. And again, it's the same rule. A and T if it is DNA replication, but since this is protein synthesis and you're using RNA and mRNA, you're using mRNA because this is, I mean, you are using RNA. This is mRNA, remember, and this is tRNA, so both of them are RNA. So they don't use thymine, they use uracil. So A and U are paired up and so on and so forth. So that if you have a codon, and that codon is CAC, like here, what the tRNA that will pair with it is GUG. So this is the codon, this is the anticodon. It's being paired, okay? And this process is now the start of the second part, which is translation. Okay? So you have mRNA, and then you are creating that into a protein. That's now the translation part, right? Now, let's continue first before we delve into some other details, right? So it's base pairing, but why is it base pairing the tRNA, sir, with the mRNA? Well, the main purpose is really this part. Sorry. The main purpose is really this part. The main purpose here is this amino acid. Okay? And if we continue playing this, then you would be able to see what's happening. So again, you have base pairs, and that's a transfer RNA. So in the transfer RNA, you have anticodons at the bottom and you have amino acid at the top. So it base pairs, right? So good, it base pairs, but what's the purpose for that, sir? You will soon see. You see, that's the purpose. Look at what's happening at the top. What's happening is the amino acid that the tRNA carries gets stitched together into a sequence. See? And then once the amino acids are stitched into a sequence, they fold together. They fold in a certain particular 3D shape, and now it becomes a protein. So, first thing first, a protein, therefore, is simply a chain of amino acids. Okay? So when you want to create a protein, you simply have to chain together sets of amino acids. And then after that, the chain of amino acid will simply fold into a particular shape and it becomes a protein. Okay? Now, that's the first part. The next part is that this amino acid here, this transfer RNA, doesn't just carry any amino acid. It has a particular assignment. So, this amino acid here is not just any amino acid. It's not just a random amino acid. It can be, it's a specific amino acid. And what determines that? It's the anticodon. So a particular anticodon carries a transfer RNA with a particular anticodon carries with it a particular amino acid. Okay? And this is now the key because if you think about it, if you look at our analogy of food items and recipe, the amino acids, therefore, are the particular ingredients in the food, right? And you have to mix the ingredients. In this case, in the analogy of food, you have to mix ingredients. In the real sense, mixing the ingredients means chaining them in a particular order. That constitutes what a protein is. Okay? And again, this is not just a random pairing because each transfer RNA carries with it a particular amino acid. Okay? So, now you can know what amino acid is brought about by which codon. So, for example, here, let me show you this video on my screen. So, for example, here, the tRNA that has the anticodon UUU. Sorry, I should have made this pink. The anticodon UUU carries with it lysine. And the anticodon the tRNA that has an anticodon CUA carries with it aspartic acid or asparagine, right? And then the tRNA that has an anticodon AAG carries with it phenylalanine, right? And in fact, you can see it here, the tRNA that carries with it that has an anticodon ACC has the amino acid tryptophan. Okay? So, therefore, this particular anticodon corresponds to certain amino acids, certain ingredients. And what's the sequence of the anticodons that are being stringed? What's the sequence of the amino acid, of the string of the amino acid? It depends on the string of anticodons. But what does the string of anticodons depend on? It depends on the string of mRNA. But where does this string depend on? It depends on the DNA. So this is why we say that the instructions for creating proteins is really from the DNA, because it's the sequence of the DNA that is read and transformed or transcripted into mRNA, and then the mRNA is again translated into a sequence of amino acids that we call a protein. Okay, but the idea here is the way it is being done is, again, the DNA will be read by the RNA polymerase. Now it creates mRNA. Some introns are removed, and the exons are the ones that are kept. Then the mRNA goes to the ribosome. The ribosome base pairs them with tRNAs, and the tRNA will simply base pair with it and then leaves its amino acid to be chained in a chain, to be chained together and create a protein. Okay? And again, I want this again to relate to your other concepts. For example, now, sir, what happens to this tRNA, sir? Now that, for example, it has attached here, it has removed its tryptophan, right? It has no amino acid. What happens to this? Guess what? This tRNA will go to the cytoplasm and then find another tryptophan. Find another tryptophan amino acid, attaches to the tryptophan, and then wait until it's called again by the ribosome. Okay? So I want you to get that idea that, because again, study hard, not smart. Why is it called tRNA? Because again, the function of the tRNA is to have anticodons here, and then to have an amino acid. And what it does is it goes to the ribosome, it detaches its amino acid so that the amino acid can be chained. And then once it has no amino acid, guess what? It will go back to the cytoplasm, find another tryptophan that is floating around in the cells, and then connect with the tryptophan again, and then wait again until it's called by the ribosome. So therefore, its job is really to transfer the protein, to transfer the amino acid from the cytoplasm to the ribosome. Gets? That's why it's called transfer RNA. And then now, you might also be asking, sir, when it has no amino acid and it finds an amino acid in the cytoplasm, why can it find amino acids in the cytoplasm? Are there a lot of amino acids floating in the cytoplasm? Yes. And again, it goes back to your digestion. So see, proteins are a chain of amino acids. And proteins is like the meat of the chicken, the meat of the pork, right? Even the mungos or mungo beans. Mungo beans has a lot of proteins. So when you eat food, the proteins from these meat materials that they have, your digestive system actually digests them into smaller and smaller pieces. And guess what those smaller pieces are? Those are amino acids. So those amino acids will now be delivered by your circulatory system to your cells, and those amino acids will just be floating in the cytoplasm, waiting for a tRNA to attach to it. So that tRNA, depending on what anticodon it has, again, if its anticodon is UUU, it will look for a lysine amino acid, and it will attach to it, and then that tRNA will just be floating until again it's called by the ribosome, and this process happens. So again, I want you to understand why it's called a transfer RNA, because its function is literally to transfer amino acids that are floating in the cytoplasm to the ribosome, so that the ribosome can assemble them into a chain of amino acids that we call a protein. But take note that the order of the chain depends on the anticodons of the transfer RNA, but those anticodons depend also on the codons on the mRNA, because again they are base paired. Remember, they are base paired. And then one step further, the codons in the mRNA depends on the bases of the DNA, because remember, it's during transcription that is what is happening. The bases from the DNA is copied into an mRNA codon. Obviously, the mRNA has to remove some of its introns and only keep its exons. But the idea is the instruction in the codon comes from the DNA. So that's why we say the protein that you create in your body depends ultimately to the sequence of DNA bases. But yes, there is a process. And the process is the DNA bases are transcribed in the process called transcription into mRNA. After that, some of it are spliced, some exons, some introns are put away, and some exons are kept. And then it goes to translation, where the mRNA goes into the ribosome. The ribosome base pairs it with the tRNAs, and the tRNA's job is just to transfer amino acids. And depending on the sequence of codons here, that also affects the sequence of tRNAs that go into the ribosome, which affects the sequence of amino acids that eventually affect what kind of protein you will be producing. Okay, so that's the idea here. So you have codons and anticodons. Now, obviously, because science is a simplification of things, obviously I simplified some of the things when I discussed. But again, I just want you to get this idea that there's a way to have a narrative in your there's a way to have a narrative in your in the way you study things, instead of just going over the and memorize things, right, without really understanding what is happening. So again, it pays to have these stories in your head. But anyways, you have here on the screen, for example, this is the obviously something that's familiar to you. This is the table of um, this is the table of codons, and this is a table of codons and anticodons. So if you notice here on the screen, UUU is lysine. So here AUG. Wait a minute. AUC. AUC. So you have a table here, but I guess what I'm trying to say is you have to be careful, because in most books we always talk about a table of codons. But take note that codons and anticodons are different. And there's also such thing as a table of anticodons. Okay? A table of anticodons. So, what I mean by that is, for example, lysine. So, the codon for lysine. So, if you have the amino acid, sorry. The codon for lysine is AAA, right? But again, because it's a base pair, the anticodon of the tRNA that brings in lysine is UUU. And the protein, the amino acid that it brings is lysine. So, what I'm trying to say is be careful, because there is a table of codons. There is also a table of anticodons, and obviously the table of codons and anticodons are different because again A pairs with U. So if you have a codon that is AAA in the mRNA, remember codons are in the mRNA, anticodons are in the tRNA. It's different. In the table of codons, AAA means lysine, but in the table of anticodons, AAA doesn't mean lysine. What means lysine is UUU, because remember, it must be AAA that pairs with UUU so that you can have lysine. So again, if I want to show this to you, see, AAA here, the codon, pairs with the anticodon UUU, which is attached to the tRNA that creates lysine. Now, in most books, what we usually use in high school is the table of codons. And I don't like that because it kind of ignores the importance of the tRNA. Because if you are only looking at the table of codons, meaning you are only looking at the mRNA and then what amino acid corresponds to that. And again, it circumvents, it takes away the importance of the tRNA. And I don't like that. I really like to understand that. I really want you to understand that the tRNA has a crucial job. Its job is to go to the cytoplasm, find some amino acids that are fit to its anticodon, bond with it, and then wait until the ribosome calls for it. If the ribosome says, AAA, we have an AAA, we like, I want a tRNA that is UUU, then the tRNA with UUU and lysine goes to the ribosome and attaches base pairs with it so that it can add its amino acid to the chain of amino acids which eventually become a protein. Okay? So, after that, I want to discuss this idea that if you have lysine, for example, and then you have lysine and then glutamic acid, aspartic acid. By the way, I just realized this is not asparagine, this is aspartic acid, I think. Asparagine is ARG. But I could be wrong with that. Again, I don't memorize these things. I don't find it important to memorize them, but I need you to understand what it means. So if you have amino acid, if you have a chain of amino acid like this, and then you have phenylalanine, alanine, and so on and so forth. If you change the sequence, like for example, instead of asparagine, you put in here tryptophan, and then phenylalanine, and then yeah, guess what? This is a different protein from this, even if they're the same apart from one. Okay? This protein has a different property from that protein. And that is how you get a multitude classes of proteins, different kinds of proteins, just because of the sequence. And again, this is also where mutations happen. For example, if it was supposed to be aspartic acid, but then you input tryptophan, guess what? This protein has a different characteristic than that one. Okay? And it could be something dramatic. Like, for example, if this was, for example, hemoglobin in the blood, what if changing this aspartic acid into tryptophan actually makes the hemoglobin molecule able to absorb more oxygen? Okay? And actually, that's what happens to people from Tibet. People from Tibet, because they have a very thin atmosphere there, they have actually evolved such that their hemoglobin protein is slightly different from the rest of the world, such that people from Tibet can actually absorb more oxygen even if the oxygen is thinner. Right? So that's the idea. So that's why you have different multitude of proteins, and that's from the concept of protein synthesis. So again, what's the narrative? The narrative is you have a secret book of recipes in the DNA, and in order to even get the instructions from that, you have to go into that. You have to go into the DNA to the nucleus and reads the book using the RNA polymerase. And from the RNA polymerase, you can create an mRNA. And that process, again, is what we call the transcription. And that transcription happens when DNA is transcripted into mRNA. This happens, of course, in the nucleus. Why does it happen in the nucleus? Because the DNA never really leaves the nucleus. It only leaves the nucleus during mitosis, when it divides into. That's why you can see the chromosomes lined up. But in the normal stages of the cell, it stays in the nucleus. So this happens in the nucleus. The second part is the translation, wherein once you have the DNA, well, there's also a kind of slight thing here. From here to here, you have splicing, where again, extron, introns are cut out and exons are kept, and some segments are spliced together, meaning they are joined together. But that's the idea. And then once that happens, this mRNA is translated into a protein, which is a chain of amino acids. And again, how does it do that? Through using the tRNA. Because the tRNA, again, has an anticodon, has anticodons on its foot and an amino acid here. And then the mRNA has codon. And obviously, how to put together mRNA and tRNA together is called through the organelle ribosome. So the ribosome is the one that attaches it here, but that's the idea. So you get from DNA bases to mRNA codons to tRNA anticodons to the proteins to the amino acids that they carry. So you have a chain of amino acids that make up the protein. So that's the idea. And this second part doesn't happen in the nucleus. It now is in the ribosome. And remember, ribosome is an organelle. And where is it found? Is in the cytoplasm. It's outside the nucleus already. So that's why, this is the reason why we kind of divide the two processes, because this happens in the nucleus, this happens in the cytoplasm. And that's why we have to call it transcription. This is translation. Okay? So that's the idea for the protein synthesis concept. So again, I want you to take in the narrative of the story, not the individual one. Obviously, you can look at table of anticodons, table of codons. You can study the table of codons and decodons, and you can memorize all of them. But again, that doesn't make sense. That's why you have a table, because we organize it in tables so that you don't have to memorize it, right? So what's the ideal is you understand the story, and then once you want to know the details, like what specific amino acid is for what specific anticodon or what specific codon, then that's when you use the table. But you don't need to memorize them or anything. Okay? So again, invest in the narrative of the story so that you would understand it better, rather than the individual snippets of information that can be memorized.